Everyone. Hello everyone. Uh, Hope you all are well. Today we will see how we predict number of disulfide bond present in any given protein sequence. So, you know disulfide bases are sometimes called disulfide bonds or SS bond. Actually, disulfide bonds are the covalent links between the sulfur atom of the two cysteine amino acids, and their formation stabilize their tertiary and higher structure of the protein. So, due to the importance of the disulfide bond in the protein folding and structural stability, the artificial disulfide bond are often engineered by the cysteine mutation to enhance the protein stability. So, today we will see how we predict the number of disulfide bond present in any given protein sequence. So, this we can use for this we can use very important tools, that is the disulfide tool, right? So, how we can use this tool and how we can interpret the result of this tool, that will clear in this video, right? To assess the tool, just we open the browser and type disulfide bond. So, just open the browser and type the disulfide by design2, right? And click this one. Click this one, right? Right? And this is the actual website of this tool. As you click this, the home page will appear like this, right? And here for interview data there are three options, right? Here for the interview data there are three options, like either you can choose PDB file, means upload the protein data bank file that must be saved on your desktop, right? So choose the file, PDB file, this file, and click on upload, right? Or you can add PDB ID, like here I am going to this PDB ID and click on get from RCSB, right? Or if they load the example of PDB, these are, or so we can click. After, as you click this, just for few seconds, like to give the results. Yes, now you can see yes, now you can see there are the these are the your interesting results. And how we will interpret these results? Actually, there are the basic informations, file information, that means these are the basic information of the protein sequence that I have selected for the PDB ID. So, right, and this is the heading, that means this this is the protein ID, PDB ID, and this is the hydrolase phosphoric disasters, right? And these are the title that is against the given protein structure of phosphate free ribonuclease A, refined at and force 26 against angstrom. Okay, it is the crystalline structure. This is 1.26 angstrom, and this is the PDB-ID of the protein, and this is the amino acid residues. This protein consists of the 124 amino acids, and this it has the single chain, and it also contains the secondary structure, the alpha helical structure. It is the 26% of it. This protein constitute of the 26% of alpha helical structure that constitute this helix constitute of the 3 helicos and 33 residues, and it also contain 76% beta seed that constitute the 4 seeds, double strand called the seed, and 14 strand, a single strand is called the strand, and it constitute of the 95 residues, right? This is module 1, and this is the beta factor. Beta factor, what is the beta factor? Actually, beta factor also called the temperature factor or displacement factor, or it also called the de Waller factors, and most of the protein raise 20 to 40 of the beta factor, right? Actually, in the beta factor for the crystal structure, the temperature factor, also called the beta factor, is given in the PDB file to measure the uncertainty or disorder of the each position, right? So now, in order to find out the disulfide bond, yes, in the left side we can see there is 3 options are available. So we can choose the options what kind of bond we want to see. Intrachain means we are interested to see the disulfide bond within the chain, and inter we can see the interchain. Interchain means you want to see the disulfide bond between 2 different chain or hetero chain or interchain, right? And this one is the build C beta for glycine. As you know, glycine causes the hinder in the formation of a disulfide bond, right? So, and here just look this. Zeta 3 angle. What is the zeta 3 angle? Actually, it is the torsion angle. That is the minus 87 degree to plus 97 degree, right? But we can choose plus minus 30 degree. So just keep it as default, and after that we press on run. It will show you disulfide linkage, right? So we press on run. After selecting all these parameters, just click on the run. That will show the disulfide linkage, right? Yes, result are coming here, and so this is the residues 1, residues 2. That means amino acid 1 and amino acid 2. This is the analysis tab. I will come on these results later. Now we can see the secondary structure. The secondary structure of the protein. So we will just click on the secondary structure tab. As you can see, the legend of this structure or the diagram legend of this diagram, here is the gold arrow that represent the beta sheet, gold arrow that represent the beta sheet, and this magenta helix represent the alpha helix. These are the alpha helix, and the gold bar, this gold bar represent the potential disulfide bond. These gold bar, these are the these gold bar represent the disulfide, potential disulfide bond, while these red bar selected disulfide bond. And this is the range of beta factor. This shows the range of beta factor, and by increasing the range, it converts blue to red and dark red. And you can see it, and you can you can also check the 3D view. So by clicking on just in 3D view tab, right, it will take for a few seconds to appear the 3D structure. Just wait. Actually my laptop is slow. Just wait. And yes, and this is coming. See, it is in the cartoon format, and you can see it by zoom in or zoom out, right? See, here is the gold bar. These are the disulfide bond. So you can see better in the wire frame. So we click on the wire. So you see here sorry, it is here is the 4 disulfide bond appear in this protein, right? And now you can see this, the disulfide bond. It is the clear. You can do clearly by zoom it and out, right? So here is the 4 disulfide bond is available. Now again we come back to the analysis tab, because here the one important option is available. What important option is available? From here you can see the mutation in the protein sequence. So to create the protein mutation, we can select any residues. For example, I select these two residues, and will click on the create and view mutant. See what will happen. Yes. So this is the original protein, and this is the mutated protein, right? This is the mutated protein. To see clear, we here also change the wire form. See, here is the 4 disulfide bond in the original, that means wild type of the protein. When we select mutate the 2 amino acids, it create the more disulfide bond. That means here we can see it is the 2, 3, 4, 5, 6. 6 disulfide bond is created, right? So thus you can see the variation of a disulfide bond by comparing the original and mutated proteins, right? And thus by using this tool we can check the number of disulfide bond appear in the desired protein sequence, and this result you can use for your thesis, dissertation or publication, right? That's all over, guys. See you soon in the next video. Thank you so much. अगर वीडियो अच्छा लगे प्लीज़ लाइक एंड सब्सक्राइब जरूर करें. थैंक यू सो मच.